à recevoir Gilda Cacavo, qui va nous présenter son travail et qui va se présenter aussi.
Oui. Bonjour et merci tout le monde pour être venu. Oui, je m'appelle Gilda Cacavo. Je suis basée à Paris. Je suis entre deux labos à Paris qui font partie de l'Institut de Pierre-Simon Laplace, donc LSCE et l'Océan. Ce sont des labos de climat, océanographie. Et essentiellement, ce que je vais faire aujourd'hui, c'est vraiment une présentation sur moi-même. Donc, c'est pas exactement une présentation didactique. C'est vraiment un peu ce que j'ai fait jusque maintenant dans mon carrière, un peu mes résultats, mes lignes de recherche et ce que je programme à faire, qui est un peu le titre de projet, et en particulier ce programme de recherche que j'ai développé dans les lignes de l'IRD. Et en plus, je vais faire la présentation en anglais. Donc, c'était juste cette petite introduction en français. Et après, je serai contente de discuter après en français sans souci. OK. Donc, je vais commencer. Yeah, English now. I want to just start by giving a bit of context. The research that I'm proposing to do is very much based on the importance of upwelling systems, which of course are these systems in which you have this cold water that rises up to the surface and promotes a lot of primary productivity. And so in this chart of global fishing over a century, you can see that these areas of eastern boundary upwelling systems have some of the largest amounts of fishing productivity. And in fact, in particular, one of the areas that I'm focusing on is the Humboldt Current large marine ecosystem upwelling system that actually accounts for nearly half of all of that fishery production that we see from all of these upwelling systems worldwide. And as with most things, climate change threatens these upwelling systems, and in particular, the Humboldt upwelling system in the following way. So if we think of what the situation is at present, we have winds that are shown by the blue arrows that promote upwelling, and this happens because of a difference between high pressure systems over the ocean and low pressure systems over the land. And for the moment, the idea is that it's relatively equitably distributed between the north and the south of these systems. However, what models project to happen in the future is that winds will be increased in the sort of poleward parts of these upwelling systems, which will increase winds and thus increase upwelling towards the poles, which will then result in a decrease in winds and upwelling towards the equator. And where we have less upwelling, we have less primary production, less productivity. And this is the risk that climate change might impose on these systems generally and on the system in the Humboldt Current to the west of South America in particular. And so this system on the west coast of South America promotes a ton of important fish species. And in particular, I'm going to be focusing on 5 of these. So the Peruvian anchoveta, the South American pilchard or sardine, South American hake, Humboldt squid, and the Patagonian toothfish. So just to give you an idea of how exploited and how much of these fish are actually caught, this is a plot from the FAO that shows in millions of tons the amount of anchoveta and pilchard that have been fished since 1950. And then in the bottom chart, just because there's sort of far fewer, we have the hake, the squid, and the Patagonian toothfish. And what you can appreciate from this chart is the fact that these catches are characterized by periods of boom and periods of bust, as well as also the recent increase in catch for Humboldt squid that we see in recent years. And so these periods of boom and bust have historically been categorized, have been caused by this natural climate variability in things such like as ENSO that drive these fluctuations in the amount of these species that we can catch. And one of the key questions that my research would like to address is how is climate change going to actually impact the already natural variability that we observe and that provokes these boom and bust periods? And so the question is, how are these exploited species going to respond to these changes on top of what we already observe with the natural variability? And so to think about species responses to climate change, we sort of have to be everything everywhere all at once. And so in some ways, you can think about how species will respond genomically. They can adapt to different conditions by, promoting certain genes via selection. There can also be a process of phenotypic plasticity, where the same sets of genes may express themselves differently in different environments. And the complexity here is that you can have, via epigenomics, processes that are actually inheritable from phenotypic plasticity, so that the actual expression of certain genes can then be inherited and influence genomic adaptation itself. And then finally, you can have species that simply change their distribution in response to existing in a less than ideal habitat with change over time. And these sort of three global ways in which we can think about species responding to climate change impacts are all of particular importance when considering early life, because this is the period in which species are generally most vulnerable to changes in their environment. And so what my research wants to do is sort of look at all of these three effects through the lens of the impact on early life. So just to give you a bit of a sense of where I've come from and what my background is, so I sort of consider myself a climate genomics researcher, generally wants to understand the impact of climate change on marine species. This term climate genomics is a term that we've sort of been coining recently, and it's just basically using genomics and other tools to understand climate change impacts on species. So the sort of broad scopes of my training, started back in the UK where I did my master's, and that's where I started sort of my training in genomics and exposure to conservation genomics and those sort of things. I then did my PhD in Italy, and that's where I started working on Antarctic fish, the little Antarctica there. And that's where I also started to integrate approaches about early life history as well, and the importance of understanding early life history to understand impacts on population dynamics. After my PhD, I got a Humboldt fellowship to do a project in Germany, and that's where I sort of changed species such that I was no longer working on just a forage species, but I started to focus on exploited species. And that's where I started to really have this interest in how could I use the methods and the questions I was looking at to inform fisheries management. So now I've just completed my second postdoc in Paris at LSE and Lausanne. And that was a project where I was now trying to bring in this modeling component. So how can we then model impacts on distributions and understand climate change impacts informed by both genomics and early life history? And next week, I'm starting, hopefully, my last postdoc, which will be specifically at Lausanne through a funding call that I got from them. And that is a project that's going to try to integrate all four of these components in a sort of pilot version of what I'm proposing to do for my long-term IRD research plan, which I'll talk a little bit more about later in the talk. So generally, up until now, I've worked on Antarctic fish. That's been more or less what I've done. And that Antarctic fish work has circulated around three main questions. What are the genetic connectivity of populations? How does early life history inform population dynamics? And how can we model climate change impacts on species distributions? And so regarding the connectivity of populations, so one of the key outcomes of the work that I had done there is the role of currents in shaping gene flow. So that work was during my PhD on this keystone forage species in the Southern Ocean called Antarctic silverfish. And what we did was we compared a set of circumpolar populations around Antarctica. And what we found was that genetic diversity, as revealed through microsatellites, had a link with populations that were connected along this Antarctic slope current shown in orange. And so basically the populations that were not found along this current, so such as the orange one at the Western Antarctic Peninsula and the purple one at the South Orkney Islands, these are the ones that we're seeing more isolation because they're not necessarily having that physical connection through the slope current system. Then more recently, the work that came out of, that's just in review now, that came out of my postdoc in Germany, is about the role of fisheries pressure on genomic diversity. So that project, following my PhD, sort of moved up the food chain fish-wise from a forage species to a top fish predator, but also approach-wise from microsatellites to reduced representation and whole genome resequencing. And there, in a similar way, we took a circumpolar data set of fish from across the Southern Ocean, and we wanted to see, well, what is the connectivity between populations and what are perhaps differences in diversity and other aspects that we can observe. And the interesting outcome that we saw here was when looking at a measure of diversity here in the form of heterozygosity, was that this particular management area, this management area 88, or the Ross Sea, which would be down here, had significantly less diversity than the other two fisheries management areas around the Southern Ocean. And in particular, what we observed was that this reduction in diversity was driven by a reduction in diversity over time. So this area 88, the Ross Sea, is the largest area of exploitation of Antarctic toothfish in the Southern Ocean. And the reason why we compared pre and post 2000 cohorts is that around the year 2000, there was a big change in how the Antarctic toothfish fishery was managed in ways to reduce illegal and unreported and unregulated fishing, and so on. And so these results said to us that not only do we see a change in diversity perhaps linked to overfishing prior to the increase in regulation, but we generally see that this area that undergoes greater fishing is also causing a reduction in the diversity pool of fish in that particular area. So that was sort of the work I've done so far on genetic connectivity of populations and different species around the Antarctic. And now with respect to early life history, so some of the early work that I did, this was during my PhD, was ways in which we can identify novel spawning grounds for different species. So here, this was work with my colleague Cassandra Brooks, that I contributed to. And the idea was that with Antarctic silverfish, that small species that we saw earlier, we know that they spawn in this western part of the Ross Sea, but they had found a bunch of larvae in this eastern part for which they had never known that there would be a spawning ground there before. And this was a relatively simple analysis of just DNA barcoding to see that indeed the larvae that we found in that eastern Ross Sea correspond to the species Plurigramma, to Antarctic silverfish, meaning that they must have been spawned there because there's no way that fish spawned here at that age would have ended up here. And so this was just a kind of clear, helpful way to come to identify that we might actually have more spawning grounds than we realized for this species in this key area of its distribution. And then on top of this, with respect to other ways in which we can inform early life history dynamics, the other work I started in my PhD and also did a bit during my postdoc in Germany as well is using otoliths. So I'm sure most of you all know, but I'll just remind that otoliths are fish ear bones. And they're typically used for aging, but we can also use them as proxies. Layers of calcium are added to the bone in a pattern over time. There's also trace amounts of elements in the ocean that are added in. And so with these trace amounts of different elements, we can correspond that to different water masses. And that allows us to sort of understand if fish came from similar water masses or different ones. And so there, I used the same samples that in the previous population genetics study, we had found no difference in genetic connectivity. These fish were genetically indistinguishable in the genetics analysis. But when we looked at the otolith chemistry and the patterns of trace elements there, what we found was, for example, that's weird that cut off like that. Anyway, what we found was that fish coming from different parts of the Weddell Sea that were genetically indistinguishable, we could actually see differences in the trace element patterns, indicating to us that these fish were born in separate spawning grounds, which gives us a whole nother level of understanding of these dynamics of the population, which just from genetics, we would have assumed that they're all doing more or less the same thing. So that's sort of the work on early life history. And now with respect to ways in which we can model climate change impacts. So the first work here that was kind of cool was the way in which we could identify how climate change may restrict prey access. So this is now back in the large Antarctic toothfish, top fish predator. And this was work that I did with a colleague, Kara Neeson, using an approach called the aerobic growth index. And so this is a way to sort of create a proxy for like metabolism and so on. And what we did was we took the aerobic growth index of Antarctic toothfish, as well as a whole bunch of its most important prey species. And the idea was then to see if with changes in different water mass properties over time due to climate change, will there be a change in the distribution of prey, and how will that impact perhaps distributions of Antarctic toothfish itself? And so what we found was that under the highest climate change scenarios, that almost 20 to 30% of prey species that were normally distributed alongside Antarctic toothfish were being reduced, particularly in depth layers between 500 and 1,000 meters. And the way we did this analysis is we wanted to consider, again, these different fisheries management sectors of Antarctic toothfish, in the Southern Ocean generally. And so the big losses that we were observing were in this East Antarctica area 58 and in the Ross Sea area 88. And what this highlighted to us is the importance of Area 48 as a potential refuge if the climate change impacts occur as there might be predicted for Antarctic toothfish and for its prey. And for example, this Area 48, this Weddell Sea, is an area where there's ongoing efforts, for example, to implement a marine protected area. So this was a kind of neat way to implement this type of aerobic growth index to try to understand how changes in prey distribution might ultimately impact any given species of interest. And so then finally here, this is work that's come out of my most recent postdoc in Paris, which is the ways in which environment drives adaptation. So again, we're here with the Antarctic toothfish, and what we were able to identify using an analysis called genotype environment association, which associates variation in adaptive genomic markers with variation in the environment, is that salinity, current, and sea ice seem to be driving changes in the variation in adaptive markers between different fisheries management areas, which are identified by the different colors that we can see here, the Weddell Sea, East Antarctica, and the Ross Sea. And in particular, salinity was the one that came out as the most significant. And this is also a paper that's under review now, but the thing that was really cool about the salinity question was, because initially, well, there's a whole bit about Antarctic toothfish early life history that I don't go into here, but the long of the short of it is that they depend on sea ice in their early life. The larvae live under the sea ice edge. They depend on special plankton that exist under the sea ice. And so you would think, based on that, the hypothesis is, well, sea ice is going to be the most important factor, right? But it didn't actually turn out to be that way. So we're saying, well, why is salinity coming out as the most important factor? Well, the thing is that these fish, they're benthopelagic, so they spawn on these seamounts near the ocean's floor. And what happens is the eggs then float to the surface. If there are changes in salinity or mixing, this could impede the ability of the eggs to reach the surface where they're protected from predators, where they can develop in an environment. And so this is where we're thinking the role of salinity in driving certain adaptive properties related to osmoregulation and buoyancy and so on might be at play. So that was the cool thing about that study. And so all of this is sort of the work I've done up until now with respect to research, but there's also been a large part of the work I've done that's been related to outside of research things. So the first is with respect to fisheries management. So as I mentioned, starting during my postdoc in Germany, when I started working on an exploited species, I wanted to sort of get into, well, what are the ways in which this work can actually inform policy? And so that's meant working with this organization called CAMLAR in the Southern Ocean. They manage all of the exploited species. CAMLAR is the Commission for the Conservation of Antarctic Marine Living Resources. And there, what I was able to contribute, the little I was able to contribute, was, for example, in the development of a new stock hypothesis for Antarctic toothfish, particularly in the Weddell Sea. But also in that identifying of the novel spawning ground. I talked about when we had found that novel spawning ground for Antarctic silverfish. And this was just one among many other pieces of data that were used to support the eventual passage in 2016 of the marine protected area in the Ross Sea, which at the time of its passage was the largest marine protected area in the world. So this is the way that this kind of work could, in its small way, go into actually affecting, policy and conservation impacts in the real world. And so then another big part of the work that I've had the opportunity to do up until now has been surrounding in what I would call international engagement. And so this has been, leadership roles in all kinds of, Antarctic, because I work with Antarctic fish until now, organizations. So some of these were related to early career scientists. So APEX, I was involved in for many years. The Southern Ocean Observing System, they also have a global version, GOOSE, as well. And then the Scientific Committee for Antarctic Research, where I'm still involved in a group that coordinates all kinds of research on Antarctic fish, which we called SCARfish. And so this work, other than being a great way to connect with colleagues all over the world, allowed me to, be able to push for the impacts that early career researchers can have. For example, we pushed for early career researchers to take roles as reviewers of IPCC reports. That was a big thing we did through APEX. And I was also quite involved in a lot of equity, diversity, and inclusion initiatives, both through APEX and through SCAR. And another group that I was involved in was related to Southern Ocean conservation. And so this was sort of like the big takeaway was the ways in which interdisciplinary coordination could actually promote conservation in real world contexts. So this is all the background. Now moving towards the future. So the idea is that we want to think about how do we connect from Antarctica to South America? What are the connections here? And so the way I see it is that there are a lot of shared questions relating to the impacts of climate change on oceanographic processes that underpin connectivity. So when we think about this in the Antarctic, we think about the southern annular mode, sea ice dynamics, stratification. And with respect to the Humboldt Current Ecosystem, there we're maybe more thinking about the El Nino Southern Ocean Oscillation, upwelling timing and intensity, as we talked about earlier with respect to potential climate change impacts, and also the potential changes in stratification. So a lot of the questions that I've been trying to address the impacts of on actual species in the Southern Ocean are very much the same as what we would want to look at in the Humboldt Current Ecosystem, which, as we saw from the very beginning, is this incredibly productive ecosystem. And so through all of the collaborations that I've developed and created over my time working in the Antarctic, I managed to get connected with this group in Peru at this institute called IMARPE, which is the Marine Institute of Peru. And my collaborators there have been working for over 20 years in genomics, stock assessments, and species distributions of exploited species in the Humboldt Current Ecosystem, which underpin all of the management of Peruvian fisheries, the researchers that are at IMARPE. Fisheries in Peru account for nearly 1% of GDP there, so it's a really important part of their economy that supports exports, employment, development. And the idea is that the types of climate genomics approaches that I've been developing and implementing in the Southern Ocean are really needed for the type of questions and management challenges that are going to be faced by Peru and by IMARPE for the species in the Humboldt Current Ecosystem. And so the idea of the project, the research program for IRD that I've developed would be to identify and project the climate change impacts on exploited species of the Humboldt Current Ecosystem, working with IMARPE in Peru, as well as working with L'Océan in Paris, which is also connected to the National Museum of Natural History there as well. And the idea would be that the project would go across 3 different axes. So a bit reflecting of the way I talked about the past research, we would be looking on the one hand through genomic analyses to identify climate drivers. What are the drivers of genomic change from climate change? Understanding the early life history components of the different species that we're interested in order to then combine the outputs of the first and the second axes to understand how species will change their distributions over time. And across all of this work would be the idea to see how can we integrate outputs of all of those different axes of research into policy. Okay, yeah, so I talked through that before actually showing it on the slide, but there you go. So that's the idea. And in particular with the distribution shift, I forgot to mention is that we're not only thinking about changes to distributions, in the present, but also thinking about how conditions in the future might change distributions in the future. So also creating these dynamic future species distribution models. Okay, so I'm going to talk us through the first axis on climate drivers. So we'll have a set of samples, tissue, otolith, and so on. And these will be used to create a set of genomic libraries, and these will be to inform genomic adaptation. And then epigenomic libraries. So when we talk about epigenomic libraries, this is also a relatively new type of approach. There's not a huge amount of studies that have worked on it. And it's this idea of selecting the methylated parts of the genome that are specifically the ones that can be turned on or turned off in different conditions. And so we would look for adaptive markers in that particular part of the genome, whereas with our general genomic libraries, we're looking all over for indications of adaptation. And so with these two sets of data, we would then perform two different genotype environment association analyses. And so this genotype, I mentioned it a bit earlier, but I'll go into a bit more detail here. The idea is that we take a set of environmental data, and so this environmental data, the plan would be to get it, from global ocean data sets, but also MARBEC has a bunch of really great regional downscale data sets if we want to go more into granular detail about conditions in the Humboldt Current Ecosystem, particularly by Peru. And what we can do is we can take the variation in whatever set of environmental variables that are both relevant to our species, but also susceptible to climate change, and compare that variation with variation in frequencies of different adaptive markers, either from the whole genome or from the epigenome. And the idea is what we would come out with are a set of environmental variables that would be identified as significantly driving those associations in the variation. And examples of outputs could be anything from temperature, mixed layer depth, oxygen, primary production, and so on. And so the idea is that this kind of work this would be one of the first things that we can get started in the project because we'd already have the samples, build the libraries, run the analyses, and get that off going right away. So now for the second axis, this is something that can sort of happen in parallel, because again, for that third axis, we sort of need the outputs of the climate drivers and the early life history components. So for the early life history analyses, there's sort of two main approaches that I would want to use to inform our understanding of early life history in the species that we'll focus on. So the first is otolith chemistry. I talked a bit about that when I talked about some of my previous results. And so the idea here, you prepare the otoliths, you then analyze them on something that's called like a laser ablation inductively coupled mass spectrometer. It's a bit of a mouthful. And there I have a collaboration with the University of Po to run these analyses. And what would come out of this is that we have a set of trace element profiles from the very center, the very nucleus of the otolith, which is representative of the first material laid down in the fish's life. And so whatever trace elements laid down there should in some ways reflect the environment that the fish was exposed to. And so with these trace element profiles from fish from various different parts of a given species distribution, we could identify whether they come from shared or separate spawning grounds. So that's approach number one. Approach number two is ovulotags. And I'll talk a little bit more about that because I'm actually here in SETS today in part because of this particular approach. So what's an ovulotag? An ovulotag combines a pop-up satellite tag with an acoustic pinger. This is actually a collaboration with Marie-Laure and others from Calavas, who we were able to get this 10,000 euro funding from Carnot Mers to try to implement this technology. Now, mind you, this is not something we could apply to all 5 species that I mentioned in the beginning, because this tag is about this big and the acoustic pinger is maybe like the size of my pinky nail, but still like a sardine or an anchoveta, that's way too small. However, 3 of the other species on the list, the Humboldt squid, the Patagonian toothfish, and the hake, would be more than adaptable for this method. And so the idea is that you insert this acoustic pinger into the ovary of the fish, into the gonad of the fish. And then the fish has the pop-up satellite tag attached to its back. And there's a constant communication between the little acoustic pinger that releases a signal every 20, 60, 90 seconds, and the pop-up satellite tag detects it and says, okay, you're there, okay, you're there, okay, you're there, every 4 minutes. And at some point, the idea is that when the fish spawns, the acoustic pinger is released along with the eggs. Suddenly, the pop-up satellite tag does not receive that signal that it's there. And after, you can program however many non-signal captures to identify that it's actually gone, that will trigger the pop-up satellite tag to then float to the surface, where it will transmit its data to the Argo satellite. And what we will then have is not only the location, but the conditions leading up to the spawning event, because this pop-up satellite tag, for all of the time that's been attached to the fish, is giving us information about depth, about light, about location, and temperature, and so on and so forth. And so actually, hold on. Great. So this is a video that we just took last week of one of the sea bass in the basin at Palavas. And the idea is that we wanted to get a proof of concept of this technology. So this is very brand new. It's from a company called Desert Star in the US. There's only one other research group that's tried to implement it. They're these Danish researchers that have been trying to implement it in the European eel. And the idea is that we want to see, number one, is it actually possible to get that acoustic pinger up into the ovary? Does it actually end up in the ovary once it's there? We're using an ultrasound, for example, to identify where we are, to see the actual little piece of metal of the pinger in the ovary. All of that we've been able to establish. The big question is, when the fish spawns, does the pinger actually come out, or does it get stuck? For example, up until now, we've already seen that we don't see any changes in, for example, development of eggs in the gonad where the acoustic pinger is, which is already a good sign, because that could, be an effect as well. So that's number one. Does the pinger actually come out? And number two, does the tag actually register that event? So those are the next sort of steps that we want to confirm. We've been sort of running trial and error with these tags over the past week or so. And the idea is, if we can establish this proof of concept, the hope would be to be able to start to deploy these tags in the field starting next year. Okay. And so, so the idea of this work, in some ways, the autolytic chemistry can sort of start right at the beginning of the project, like the genomics work. And the ovula tags, as I mentioned, if all goes well, they can maybe be released into the field starting next year. And that would be work that would be with MARBEC to release them on their field deployments. So now the last axis. So how do we bring it all together? So first, there would be a component of Lagrangian modeling with my colleagues at L'Océan. So once we've identified sort of the shared and separate spawning grounds, as well as perhaps for some of our species, even locations of spawning because of the ovula tags, we could then use that to feed into models of larval dispersion using Lagrangian modeling. So we take that idea of where the early life stages are dispersing, and we combine that with the drivers that we've identified of adaptive genomic variation in our first axis to create a not only genomically informed, but hydrographically informed species distribution model. First, for present distributions. Then for the future, what we would do is, again, taking those outputs from our genotype environment association, what are the key environmental variables driving adaptation? And we combine that with variation in future conditions from both regional downscaled models as well as CMIP6 datasets. And we can use these two pieces of information to perform an analysis called genomic offset. So what genomic offset does is it quantifies maladaptation to future conditions. And with this genomic offset, we would then hopefully be able to build a sort of future species distribution model. Where are individuals across a distribution going to be better or less well adapted to future conditions? So that's more or less all of the research parts of the work. Now we have the policy integration parts. So this is obviously something that would be a key, that my partners in Peru would be a key part of in order to engage with national fisheries managers, not only in Peru, but of course the Humboldt Current Ecosystem is not just in Peru, it's also in Chile, it's also in parts of Ecuador. So it'd be key to work with those colleagues there to connect with fisheries managers in all these parts of the system where the outputs of this work could be useful. And there's also this regional fisheries management organization called SPRIFMO in the South Pacific that has 17 different countries, of which, Peru, Ecuador, and Chile are also members. And so this could also be a key organization to engage with, again, to get the outputs of the work into actual impacts. And so examples of the type of policy interests that could come out of the different research axes, once we know the climate drivers, this could be something that could be used to potentially adjust climate catch limits, inform spatial planning, where we see that the most important drivers of adaptation are occurring. The results from our early life history analyses can help to inform, yes, also spatial planning, but also potentially stock assessments. And then finally, our species distribution models would contribute to climate responsive management strategy. And this is something that the goal would be to have like a continual engagement throughout the period of the project. And so just to sort of recap, like who is involved, because this is a really big project with lots of people that are contributing. I know a little bit about everything, but I'm not an expert in everything. So of course, IMARBEC. At the Museum of Natural History, I have a lot of colleagues that have done this type of genomics work as well. There's also colleagues at IMARBEC that are involved in that portion as well. For the autoliv chemistry, as I said, that's a collaboration with the University of Po. And of course, we have the IFRAMER collaboration with the ovula tags. Then for the distribution shifts, there's a lot of great hydrographic modelers at L'Océan who I would be working with. Climate models would be work that I would collaborate with people from my previous lab, LSC, but also IMARBEC. And then finally, with the species modeling components, there's a lot of great work with my colleagues at the Museum of Natural History that have done this sort of climate integrated species distribution modeling. And then, of course, for the policy integration, L'Océan has a huge amount of experience of that, as well as IMARBEC to work with those national managers. And so you might also ask, well, why L'Océan? Like, there's many places that this work could take place. So L'Océan, to be fair, with most institutes and labs, I think, is keyed into a lot of these global initiatives, such as like the UN Ocean Decade and so on. But also they have a huge connection with the Global South and with IMARBEC in particular. There's a lot of researchers that I'm working with at L'Océan that have had ongoing connections for decades with the groups in Peru and with fisheries management work generally. In particular, the people at L'Océan have really developed have contributed to the development of these regional downscaled ocean models in the Humboldt Current Ecosystem, like Alice Pietri and Vincent Echevin, who are two of my collaborators on the project. So that's a really key component of why it's really helpful to have this work happening through L'Océan, is because of those existing connections. And also because of the fact that L'Océan is connected to the Museum of Natural History in Paris. They have this great genomics lab where all of the, for example, genomics components of the work could take place. And also there's teams at the Museum of Natural History. Aquatrend are a lot of the specialists in species modeling, and the DIVA teams are specialists in this sort of climate genomics, genotype environment association sort of work. And so just to give you an idea of where I might be one day. For the past few years, I've been trying to develop a project for CNRS, which is slightly different from this project, but it's along the same sort of lines. So that didn't work out the past 2 years. I applied again this year. Note that the section numbers have changed because they changed the sections in CNRS for some reason last year. So don't know yet what's going to happen there, but we'll see. Haven't announced things yet. Last year was the first year I applied to IRD to 2 different sections, CSS3 and Inter-CSS. So I was pre-selected for the CSS3. I presented, but I was not shortlisted. I'm applying again this year with the revised version of the project. I'll actually be leaving for Peru in about a week or so. Don't still have plane tickets yet, but that's how it goes when starting a new position. With my colleagues at IMARBEC to revise the project and resubmit that later this year. And on top of that, the Museum of Natural History is opening up a maître de conférence, a lecturer position this year, which I will also be trying for. So lots of possibilities. The research projects are slightly different. The CNRS and the Museum of Natural History would be more Antarctic. There's the IRD work is what I've shown you here, which is the Humboldt Current Ecosystem. But again, it's a very similar outcome. It's just a different place where it's going to end up. So in sum, as a climate genomics researcher, my big goal is to understand climate change impacts on marine species, and in particular with this research program, exploited species in the Humboldt Current Ecosystem. And so again, this work is possible because of a huge group of partners, my partners at MARBEC, and then across all of France. So thank you. Maintenant, on peut parler en français.
Bon, ben, est-ce que vous avez des questions? On prend d'abord dans la salle, peut-être. Sophie.
Merci. Merci beaucoup. Tu as fait une excellente présentation, très, très efficace. Bravo. J'avais une question plus précise sur l'aspect ovulotag. Non pas sur la technique, mais sur si tu pouvais nous détailler un petit peu l'hypothèse que tu fais, parce que tu es passé vite, mais tu veux regarder des petits pélagiques, et puis par les contraintes techniques, on comprend très bien, tu utilises cette méthode sur d'autres espèces, le merlu ou le giant squid, le calmar géant. Mais tu nous as pas expliqué comment tu utilises cette information-là pour ton panorama général, parce que c'est quand même des écologies assez différentes. Les spawning grounds, c'est pas forcément les mêmes. Enfin, je sais pas. Comment tu vois ça?
Oui, alors, mon idée pour comment utiliser les sorties des ovulotags, c'était pour identifier des zones de pente pour les simulations de Lagrangiens, essentiellement. Donc, ça va informer les simulations de Lagrangiens où on va sortir les particules. Et ça, après, informerait comment on développe les modèles de distribution où on dit qu'il y a plus de probabilités ou moins de probabilités à base de ça. Donc ça, c'était comment j'avais imaginé le voyage de l'information. Est-ce que ça, c'était ta question?
Ouais, c'était comment tu fais le transfert d'informations squid et merluche vers petit pélagique, et enfin, quels sont les risques en fait?
Les risques?
Les risques de regarder un jeu d'espèces pour faire des inférences sur ce qui peut se passer pour d'autres espèces.
OK, merci. OK, j'avais pas compris. Tout ça, c'est une espèce, une espèce, une espèce, encloistrée dans un... Donc, on ferait ça pour le merlu. Après, on fait une distribution d'espèces pour le merlu, mais on ne présume pas que ce qui se passe avec le merlu va nécessairement impacter la légine, par exemple. Donc, pour le moment, c'est vraiment une focalisation espèce par espèce par espèce. C'est tout à fait vrai que c'est possible dans des autres types de modèles de développer quelque chose qui intègre les espèces, mais c'est pas nécessairement pour cette première partie de projet que j'ai en vision, parce qu'en plus, pour les parties génomiques, pour avoir une compréhension de où on a le plus de pression d'adaptation dans les différentes zones. Mais ça, c'est vraiment quelque chose qu'on peut que définir pour une seule espèce à un temps. Donc, tout ça, c'est un bon point peut-être à renforcer, vu que je parle de beaucoup des espèces, mais je parle pas de comprendre l'interaction entre les trois ou les cinq. Bien sûr, il y a des interactions, mais ces types d'analyses doivent être pris une espèce par une espèce. Et la façon qui se construit, on peut pas nécessairement à ce stade. Il faut développer les méthodes, les modèles pour une espèce, et après, on peut potentiellement commencer à penser des interactions. Ça, c'est vraiment individuel.
Arnaud?
Bonjour, je suis Arnaud Bertrand, on avait interagi l'année dernière.
Ah.
Merci pour ta présentation. Bon, j'ai pas mal de commentaires, mais peut-être qu'on pourrait aussi discuter à un autre moment pour pas embêter tout le monde. Sur les drivers physiques, tu parles beaucoup d'ENSO. Alors, c'est sûr qu'à haute fréquence, c'est important, mais j'imagine que tu as regardé les travaux de Renato Salvatecci qui sont basés sur la paléo. Et en fait, le Humboldt, il est quand même soumis à énormément de de modulation à différentes fréquences. Et en fait, le Humboldt, il est très productif que depuis 100 ans. Avant, il était beaucoup moins productif. Et donc, les travaux, notamment avec Vincent, montrent que on peut avoir des vrais régimes shift et que dans quelques années, on pourrait retomber sur un autre état très, très différent. Donc, je pense que Enso, autant il a des impacts à haute fréquence, autant on n'a jamais pu montrer qu'il était le driver des changements de régime. Je pense vraiment qu'il faut que tu repenses un peu, de façon un peu différente, ta problématique climat, en regardant les autres échelles de changement, et vraiment les travaux de Renato sont très importants.
Mais ça, c'est un super bon point, hein. Merci.
Après, bah, il y a aussi l'oxygène. Tu en as un petit peu parlé, mais c'est vraiment un driver fondamental. Et je pense justement qu'avec toutes tes approches génomiques, otolithes, etc., c'est quelque chose que tu devrais pouvoir voir. Euh, le merlu, ils sont tout petits. Je ne sais même pas si tu pourras mettre tes tags parce que justement, ils ont été tellement surexploités qu'ils sont vraiment petits.
Ah oui,
Quand on les pêche, ils dévaginent souvent la vessie natatoire. Donc, je ne sais pas si tu arriveras à mettre tes tags sur les merlus.
ok, intéressant.
Mais par contre, ils sont très intéressants pour la génomique et ton approche, puisqu'ils ont été tellement surexploités que maintenant, enfin aujourd'hui, je ne sais pas, mais il y a quelques années, seules les femelles qui arrivaient à se reproduire à 2 ans pouvaient survivre, enfin se reproduire. Donc on a eu une sélection très très forte. Est-ce que c'était de la plasticité? Est-ce que c'était... Donc là, il y a quelque chose.
Oui.
Et à ce sujet-là, il y a une espèce qui pourrait être très intéressante pour toi, alors qui n'est pas exploitée, qui est la Munida, le squat lobster,
Ok.
Pleuroncodes monodone, qui est une espèce de galatée pélagique, un squat lobster, qui est benthique au Chili et dont la population s'est étendue vers le Pérou vers les années 2000 et qui est devenue pélagique. Et donc, pareil, est-ce que c'est de la plasticité? Est-ce que c'est de une sélection? Enfin, et c'est devenu un aliment très, très important pour aussi bien les mammifères marins que beaucoup de poissons. Donc, c'est une espèce qui pourrait être intéressante pour... Enfin bon, j'arrête là,
Non, mais
mais n'hésite pas.
je suis toujours partante de considérer des autres espèces aussi. Et je pense, tu lèves au moins point sur les faisabilités des ovulotags sur les merlus, par exemple. Euh, Parce que c'est vrai que les ovulotags sont super chouettes et je suis ici pour ça, mais ça reste une petite partie du projet et pas nécessairement fondamentale pour tous les aspects. Parce que bien, on aura peut-être jamais la possibilité d'impliquer ça, par exemple, pour l'anchoveta ou pour le sardine. Et donc, l'idée, c'est qu'il faut quand même avoir des autres façons d'informer le jeune histoire de vie pour éventuellement informer les modèles de distribution d'espèces. En plus, les ovulotags, ils sont pas donnés. Ils sont 2000 balles chacun. Et pour pouvoir avoir une certaine quantité, pour pouvoir implémenter aussi l'utilisation plus régulier de cette technologie à l'IMARPE, mais ça, c'est une grande barrière financière aussi. Donc, c'est intéressant, c'est informatif, mais c'est pas nécessairement clé pour pouvoir éventuellement développer les modèles qu'on veut, parce que bon, ça sera pas nécessairement faisable physiquement ou même financièrement pour toutes les espèces.
Juste un tout petit détail.
Bien sûr.
Peut-être évite de dire pendant le concours que l'océan est spécialisé en fisheries management. Surtout Vincent et Alice qui sont très très bons, mais c'est pas du tout leur spécialité.
Leur truc, mais
Donc,
les autres font à l'autre.
ta place,
Et oui,
j'ai de dire ça le jour du concours, parce qu'ils peuvent penser que c'est pas le labo le plus spécialisé en ça.
oui, il y a aussi, bon, je pense quand je parle de ça, je parle aussi des connexions avec les labos au muséum qui font par exemple toute la gestion de pêche dans l'Antarctique. C'est des gens avec qui je travaillais, donc oui, mais c'est vrai, il faut être sûr de ne pas donner une impression que ça c'est tout ce qu'ils font.
D'autres questions? Ah, vas-y, je vais.
Sophie, exactement. Merci pour ta présentation. Est-ce que tu peux donner un peu plus de détails sur ta partie du coup 3 dans ta projection? Ou comment est-ce que tu couples tes modèles pour déterminer ta maladaptation à l'environnement futur, entre guillemets. J'arrive pas forcément trop à voir comment ta génomique rentre dans tes modèles de distribution d'espèces, par exemple.
Oui, donc l'idée, c'est que c'est un peu différentes façons de visualiser distribution. Donc, on peut penser de distribution physique où on va la probabilité d'avoir des poissons, mais on peut aussi penser d'une distribution de cette maladaptation. Et donc, ça, c'est le génomique, ça, c'est ce que ça porte, cette analyse genomic offset. Ça essaie de quantifier à quel point les fréquences de marqueurs qui existent maintenant et qui sont associées avec certains aspects environnementaux, avec les changements qu'on voit, où on va voir une déconnecte entre les conditions environnementales et les fréquences de marqueurs qu'on observe maintenant. Et donc, essentiellement, on aura en carte des zones avec plus de maladaptation et moins de maladaptation. À ce stade, comment ça converte entre, oui, plus de maladaptation, mais est-ce que ça veut dire moins de poissons? Ça, c'est pas nécessairement quelque chose qui on peut tout de suite montrer, mais on peut montrer où il y a la distribution de maladaptation. Après, on peut dire, façon classique, à base de association d'abondance et caractéristiques environnementales, où on a cette prédiction d'abondance. Et après, on peut comparer entre les deux et faire des inférences, mais il n'y a pas nécessairement une façon quantitative d'aller de, oui, maladaptation au impact sur l'abondance. Il faut beaucoup plus des expériences sur les terrains pour vraiment convertir entre ces deux différentes valeurs. Mais c'est tout à fait vrai, c'est deux différentes façons de penser, visualiser les trucs, mais qui communiquent pas nécessairement entre eux.
Du coup, pour être clair, quand tu parles de maladaptation, tu parles de traits morphologiques, de caractéristiques dans le développement. Enfin, c'est quoi que tu appelles maladaptation? Comment tu fais le lien entre ta génomique et ton phénotype, en fait?
Oui, donc il n'y a pas nécessairement besoin d'avoir une connexion entre les deux parce que c'est vraiment à base d'un truc statistique. Donc on voit une connexion entre la variation dans le facteur environnemental, on peut dire oxygène, et ce changement d'oxygène explique ce changement de fréquence des marqueurs qui sont dans des parties du génome en train d'être sur l'adaptation. Et l'idée, c'est que on peut projeter avec les changements projetés dans l'oxygène, comment ça va être distribué par rapport les types de génotypes qu'ils ont associés avec ça en ce moment. L'idée, c'est que on peut, bien sûr, par exemple, pour les présentes, on peut faire cette analyse, Genotype Environment Association, et on peut dire, OK, on a ces marqueurs dans les génomes qui sont associés avec l'oxygène. Alors, qu'est-ce que c'est les trucs-là? On peut faire une analyse, et c'est ce qu'on a fait, par exemple, avec la légine. On a vu que les parties de génome qui sont associées avec la salinité et qui sont sous adaptation, ils sont associés avec des processus de osmorégulation, des récepteurs de calcium et tout ça. Et donc, l'idée, c'est qu'on peut faire l'hypothèse que c'est des changements dans ces aspects phénotypiques qui ont poussé par salinité qui provoquent cette association. Et donc, c'est pas nécessairement que... mais on n'a pas nécessairement besoin de définir qu'est-ce que c'est le résultat phénotypique pour quand même faire l'association. Mais c'est quand même quelque chose d'intéressant parce que ça explique pourquoi on observe ça. Et c'est quelque chose qu'on a fait avec l'étude avec les légines et qu'on peut tout à fait faire aussi pour les autres trucs proposés. Mais juste d'avoir l'association et de dire, OK, dans cette zone-là, on prévoit d'avoir plus de maladaptation. Alors, est-ce qu'il y a un intérêt donc de gérer cette population d'une façon différente à base de ça, sans devoir expliquer, mais l'association, l'adaptation sur ce système, c'est à cause d'un truc de respiration, etc., etc. Donc, c'est pour ça qu'on peut j'arrête là, parce que c'est vraiment, alors, qu'est-ce que c'est l'information dont on a besoin pour faire une question de gestion? Mais c'est tout à fait vrai que on peut plus, on allait plus loin et vraiment commencer à avoir une explication mécanistique qui va vers la phénotype pour pourquoi on observe ces associations.
D'autres questions? Peut-être en ligne?
Est-ce qu'il y a des questions en ligne?
Éventuellement une autre question.
Vas-y.
Est-ce que tu pourrais réexpliquer un peu ce que tu as fait sur les gènes, il me semble, avec les proies, les sortes d'over... Enfin,
Ah oui.
l'explication avec les proies. Pour moi, j'ai compris de l'overlap, mais je ne suis pas exactement sûre de ce que vous avez calculé pour les différentes proies. Donc,
Oui.
un tout petit peu si...
Est-ce que
s'il y a
je
le temps.
peux retourner? Je juste retourne au diapo, comme ça, on a notre contexte. Voilà. OK. Non. OK. Donc, l'idée avec cette étude-là, c'était d'utiliser ce truc qui s'appelle the Aerobic Growth Index, ou AGI, et c'est calculé à base de la consommation d'oxygène. Et donc, il y a un calcul particulier pour le faire qui est dans le papier. Et l'idée, c'est qu'on calcule cette AGI pour... attends, si je peux aller en arrière. Pour...
Tu repartages ton écran.
Ah, pardon. Oui, c'est vrai. Merci. Share. OK. Merci beaucoup. Est-ce que c'est là? OK, je réessaie, pardon. OK, ça marche bien. Et donc, l'idée, c'est qu'on fait cette calcul. Donc, le premier travail, c'était un peu de voir, alors, qu'est-ce que sont les proies plus importantes pour les gènes? Après, on fait cette calcul AGI à base de l'oxygène avec chaque espèce. Et après, on faisait des cartes de distribution de chaque espèce. Et on a intégré ces données pour on peut comprendre à base de les besoins métaboliques de chaque espèce, comment sa distribution va, si les distributions maintenant sont représentatives à ce dont elle a besoin pour ses besoins métaboliques, avec des différents scénarios, comment ça va changer et comment le overlap entre la légine et ces espèces de pois va changer. Bon, je ne sais pas si ça a mieux expliqué. Pour les détails comment c'est calculé, tout ça, je te recommande d'aller au papier parce que c'était pas vraiment moi qui a fait, c'était notre autre co-auteur Anne Moret vient d'un labo en Suisse qui ont beaucoup utilisé ce type de métrique pour analyser des questions similaires dans différents systèmes. Et Cara Neeson, c'est plutôt océanographe biochimiste. Et donc, et moi, la personne des gènes, on s'est mis ensemble pour essayer de l'appliquer ici. Mais c'est assez chouette comme métrique. Et bon, quand on parle, par exemple, de l'intégration de plusieurs espèces avec un sens de modélisation, ça, c'est un exemple de où on peut potentiellement faire ça.
Super, merci.
Merci beaucoup pour ta présentation.
Merci à vous pour votre attention.
Merci tout le monde et à la semaine prochaine pour entendre Cortez qui présentera ses travaux de postdoc avant de partir. Voilà.
J'aimerais bien parler avec toi. Tu es là encore aujourd'hui.
Oui.
Pourquoi la semaine prochaine?
Ah, oui. Oui, je suis.
Ciao.
Tant que.